Right, so good afternoon again, guys. We're starting the new module of transport plants. I'm happy to see us here that are safe. And please remember to continue to pray for students in the western end of the island who are not able to go to school anytime soon. Right, several of our students had their schools damaged, their homes damaged, and they're literally starting over from scratch. So I want you to remember that and really treat your education as really a privilege right now, right? And do the very best you can do. Now, as it concerns the transport in our plants, we had looked at transport in humans, and one of the differences with our plants is that our plants are multicellular organisms, just like humans, and so they can't depend on getting just using simple diffusion alone to get their nutrients, right? So they do need a transport system to carry nutrients to all their cells, just like how we as humans needed that. So in plants, though, their transport system is more simplified, and they need three things, really, three organs in their transport system, which are the leaves, the roots, and the stem, right? So I'm going to give you a chance to copy this. And so they're still trying to meet their needs, just like how we humans are meeting our needs with our transport system. Except that for the plants, they are not that complicated as humans, so their transport system is not as complex. Right? So go ahead and make notes of that. So they have the leaves, the roots, the stem for the transport system for a plant. So guys, so let's continue. So I know some person just came. I hope you took a photo. That's you, because I'm gonna be scrolling away. All right, so there are vessels in plants similar to how we have vessels in the human body. So there are plant vessels that are similar to human blood vessels. However, unlike humans, plants do not have a pump. So we have the heart, which was our pump. They don't have a pump. And the reason why they actually don't need a pump is that plants are not as complex as animals and they don't carry out as many metabolic processes. And so they really don't need a pump, right? Like we needed the heart to push the blood so we could get nutrients to our cells very quickly. But plants, they can get their nutrients at a slower rate because they are not as complex. They don't carry out as many processes as we do. All right. So what I'll tell you about next would be what are the vessels that are inside of our plants. So we're going to learn about two types of vessels that our plants use. Remember, if you just joined, this is week 7. Just snap a photo just in the event that I have to scroll away. So what I'm telling you about the vessels that our plants use, they have their own vessels, like how we have blood vessels. So plants have a more simplified transport system than what we have as humans. And their transport system has two main roles. The transport system has to transport water and minerals from the soil to the leaves, right? So that's one thing that the plant's trying to transport, water and minerals. And then it's also transporting sugars that are made in the plant. So those are the two things that plants are trying to transport, water and minerals and sugars and amino acids. So remember that the plant transport system has the leaf, the root, the stem. So of course, I'm going to be telling you about the three areas, leaf, root, and stem. And then you've learned that the plant has two types of vessels. So the vessels that transport water and minerals are known as xylem vessels. And those that transport the sugars and amino acids are the phloem vessels. So we're going to be learning about these vessels as well as how the leaf, the root, and the stem assist, right, in the whole process of transporting plants. And we're going to actually start off with a leaf because the leaf is the one that is most familiar to us. So we're going to start off our discussion about the leaf. So the reason why plants are really, well, one of the main reasons why they need water is because the leaf needs water in order to carry out the process of photosynthesis. So the leaf is the organ that carries out photosynthesis. And for this process, which creates sugar, we require three main things. The leaf requires carbon dioxide, sunlight, and water. The sunlight is easily obtained from the sun, so the leaf can easily get sun through the upper epidermis, sunlight through the upper epidermis. Carbon dioxide comes into the leaf through the lower epidermis, through the stomata. But water is the more difficult thing for the leaf to get because water has to be taken from the soil and then has to be transported up to the leaf against gravity. So of the three things that the plant needs in order to carry out photosynthesis, water is the most challenging one for the leaf to obtain. And that's why we're going to really focus on how we're going to get water up to the leaf. So right, so guys, we're looking at the leaf. So we're saying that the leaf is the main organ that we are trying to get nutrients to in the plant, right? And we're going to see that the leaf, there are two ways that we're going to learn about the leaf. We're going to learn about the external surface and the internal surface. And whereas they're not going to really force you to know how to draw the external one, internal one, yes. All right. So I'm going to show you the external surface of the leaf. And then I'm going to get the simplest version I can give you for the internal surface. And so you see where we are actually trying to transport the nutrients to. All right. So the leaf is the main organ that we're focusing on for now. So if you should look at the external surface of a leaf, guys, then you can see the tip of the leaf right there. So then there's a central vein in the leaf known as the midrib. The external margin of the leaf is there. And then we have smaller veins. The leaf blade itself is known as a lamina. And the stalk that connects the leaf to the plant is known as a petiole. All right. So you need to be aware of it, but I'm not going to force you to draw that. Okay. But what I need you to know is to have a diagram, internal diagram of the leaf. Just pick up that. I'm trying to find a nice simple version for you. No, you're not drawing this one. You're not drawing the external surface unless you want to sketch it. I'm going to give you the internal structure in a moment. I'm trying to find a nice simplified version for you. Right, so I'm getting one, but so as I said, don't worry about the external surface. You just need to be aware of it. The internal one, no, this one has probably some watermarks on it, but we'll still work with it. Right? Because apparently I was supposed to pay for the diagram, but right. So let me just make this bigger because the internal structure we need to know. Okay. So I'm going to ask you to ignore the watermarks from Shutterstock. All right, let us focus now on the internal structure. So if you really should get a leaf and actually put it under a microscope, guys, this is actually what you'd see. Even though the leaf looks so flat and so thin, it actually has multiple layers inside of it. All right. So as I said, ignore the Shutterstock watermark. So the first layer, you're going to see, well, if you should see it under the microscope. So this is under the microscope. Clearly, we can't see this with the naked eye. All right, so under the microscope. Now, the first layer would actually be a cuticle. Sometimes it's called a waxy cuticle. Beneath that, we have a layer of cells that are running horizontally, and those cells are from the upper epidermis. Beneath the upper epidermis cells, we have another layer of cells, but those are stacked vertically, known as the palisade mesophyll cells. These cells have a lot of chloroplasts. So these have a lot of chloroplasts in them, all right? So notice that there were no chloroplasts in the top cells, the upper epidermis cell. As I said, ignore the Shutterstock watermark on the diagram, right? So they're watermark their diagram, ignore that. So these the upper epidermis cells, they don't have any chloroplast in them. The palisade mesophyll cells are packed with chloroplasts because they are the ones that are going to do most of the photosynthesis. Beneath that layer now, we have some other cells known as the spongy mesophyll cell. They have few chloroplasts. And notice that they are not as tightly packed as the ones in the palisade mesophyll. They are more loosely packed because there are a lot of air spaces between them. So we don't want them in the spongy mesophyll to be packed. Then now we have in the spongy mesophyll layer, we have a vascular bundle. So we're going to call this vascular bundle. A vascular bundle now has the vessels of the plants. So in this vascular bundle, to the top, we have xylem vessels. To the top and to the bottom, we have phloem vessels. And it's sometimes we call a leaf vein or a vascular bundle. Then now the bottom layer of the leaf has the low epidermis cells. They don't have any chloroplast either, right? And then in the low epidermis, we have some sausage-shaped looking cells. They are known as guard cells. And then between the guard cells, we have some pores, which are known as stoma. All right, all of this is important. Every chemistry student, biology student has to know the internal structure of the leaf. Of course, you don't have to draw every single minute detail. You're sketching it, right? Sketch. Well, you need to sketch it. So I'm going to give you a chance to do that. Ignore the watermarks, Shutterstock. And get down the details. I'm going to give you about 10 minutes to do that.
Hi, good afternoon. Just a quick question. Where we have copied the first diagram of the leaf, which was the external.
No, I never needed you to copy that.
Oh,
I just want to be aware, but this one definitely I want you to copy.
oh, fine, thank you.
All right, guys, I'm going to give you 4 more minutes, and then we're going to discuss well, the main layer that does photosynthesis would be the palisade mesophyll layer, right? The photosynthesis is mainly carried out by cells in the palisade mesophyll layer. And as we said, the process requires sunlight, water, well, sunlight, carbon dioxide, and water. And I would say the process requires that sunlight, carbon dioxide, and water are supplied to these cells. So photosynthesis is mainly carried out by cells in the palisade mesophyll layer, and for them, they need to get sunlight, carbon dioxide, and water. So the sunlight is not a challenge, as we said before, because that can easily pass. So say, for example, this was the sun. The sunlight can easily pass through the upper epidermis, right? I should have mentioned to you that these cells are transparent. So can you just please make a note beside the upper epidermis cells that they are transparent, please? So that would explain to you why it's not an issue that as soon as the sunlight falls on the leaf, the sunlight is going to pass through the upper epidermis cells and easily get to the palisade mesophyll cells because these cells are transparent. All right. So sunlight is not an issue for the palisade mesophyll cells to get. So you don't have to draw this on your diagram, all right? Other thing now that they need is carbon dioxide. So the carbon dioxide now is not hard to get either because that's in the atmosphere. So what happens is that the carbon dioxide can pass through the pores, through the stomata, and it will actually go through. Let me see if I can put a pathway. So the carbon dioxide molecules that are in the atmosphere, they can come up through the pore and they can just keep going through the air spaces until it gets to the palisade mesophyll cells. So the carbon dioxide is not hard for the palisade mesophyll cells to get either. They get that through the pores in the lower epidermis. Now, the trickiest thing now is the water. The water now that the palisade mesophyll cells need are going to come from the xylem vessels, right? So the xylem vessels now are going to bring forth the water. But this water here now is coming from all the way in the soil. So remember, the leaves are on the trees and they're high up, depending on how big the trees are. And here it is now that the greatest, or the most challenging resource that we need to get to those palisade mesophyll cells would be the water. because the other two are fairly easy to obtain. So how now we are going to discuss how are we going to pump, well, not pump, how are we going to get water to travel from all the way that is in the soil, all the way to the leaves, which can be meters high. There's trees that are so tall, especially like in canyons or forests, can have extremely tall trees. And remember, we stated before that the plants don't have a pump, all right? And I'm going to just give it to you, give you a perspective for you to understand just how remarkable this transport system in the plants are. It's a remarkable system. You can have trees which are much taller than a 2-story building or 3-story building, 4-story building. You can have trees as tall as a 4-story building, right? And these trees are able to get water from the soil all the way up to the leaves at the very top, which can be as high as 4 stories. And they're able to get this water up without the use of a pump. And they are going against gravity. So imagine in our tiny bodies, we have a heart that has to pump blood to get the blood to go to like the fingertips and our toes, etc. And the plants can move water a much greater distance without a pump. To even give you some more perspective as well, even like firefighters or even for the apartment buildings that are being constructed, it is told to the developers that the National Water Commission does not provide water beyond the 2nd floor of the building. So the water pressure that they are giving, they are not guaranteeing you that that water pressure can pass the 2nd floor. So these all these apartment complexes which are higher than 2 stories, they have to have additional pumps in the actual development plan to pump water to the higher floors, right? So they need a pump. So now just imagine plants going higher than 2 floors and they're not using a pump. So that's what we're going to focus on, like what mechanism could our plants be using to get the water to travel to these great distances without a pump, right? So we've already looked at the leaf. All right. So there's a little bit more I want to tell you about the leaf, because as I said, this topic is when you're going to learn about it. And when we get to photosynthesis again, you will learn some more about the leaf. So photosynthesis is this process. Let me just make sure I mention it from now. You would have probably learned it in fourth form, but if you didn't do it in fourth form, let me mention it here. So photosynthesis is this process by which the plants combine carbon dioxide molecules with 6 water molecules in the presence of sunlight and chlorophyll, and they use that to make glucose. And oxygen is a byproduct. So when I keep making reference to photosynthesis, this is what I'm talking about. The plants being able to make glucose, and that glucose that will be made now will go to the other vessels, which are the phloem vessels, and then the glucose will be transported to all parts of the plant. Like the parts of the plant that cannot do photosynthesis, like the roots, they are going to get the glucose that the leaves made. All right. So I'm just going to mention for you the parts of the plant and their function, even though we have the leaves, even though we have seen that already. And then we're going to get back into discussing this mystery of how the plant gets the water to travel these high distances. Right, so we have a little more writing to do. We're going to be looking at the internal part. So we just saw in the diagram. We're going to be describing what each of these parts, what they do, okay? Just a moment. Let us just make a note of the parts. I assume everybody had finished copying the diagram, right? All right, so let's just make a note of the internal parts of the leaf. So the epidermis, we saw that first, right? We have an epidermis to the top, so we had the upper epidermis and we had the lower epidermis. And that's a protective outer layer on the top and bottom of the leaf. And the upper epidermis had a waxy layer on it, and that was to prevent water loss. The cuticle was the layer on top of the epidermis to prevent water loss. And then we got to the mesophyll layer, which we had the palisade, mesophyll, and the spongy. Palisade being very tightly packed, column-shaped cells at the top of the leaf containing many chloroplasts. The spongy mesophyll cells were irregularly shaped with a lot of air spaces between them to allow for gases to move easily for photosynthesis. All right, so go ahead and make note of these layers and we continue. So we have other parts, no? So I'm just gonna do the other remaining parts of the leaf. So the stomata would be the tiny pores on the low epidermis usually. The only time you might see stomata in the upper epidermis, guys, is if it's a plant that lives on the surface of the water. You know, you have like the water lilies and so on. So if they live on the water, then they're going to want to put the pores in the upper epidermis because the low epidermis would be touching the water. But no, usually it's in the low epidermis, the stomata. Those are pores. Stomata is plural, stoma is singular. And that's where we get carbon dioxide coming in, and the oxygen that we made is able to leave. And we have the guard cells, which were those sausage-shaped cells we saw in the lower epidermis. They control the opening and closing of the pores. So they will control how much gas gets exchanged, and they also control how much water is lost by the plant. And then we saw xylem and phloem. The xylem were the vessels that transport water and minerals from the roots to the leaves. And the phloem were the ones that would transport the sugar produced during photosynthesis to the other parts of the plant. All right, so you can go ahead and copy this when you're done. I'll actually show you a video so you can visualize this in 3D. And then the second half of our class is going to be dedicated to seeing how we're going to get that water to travel up all the way to the moon. I'm gonna give it 2 more minutes, guys. More minutes, guys, and then I want to show you videos to do the parts of the leaf that we just learned about. The prelude into water transport. Okay, so if everybody's finished copying this. Everybody finish? Guys at home, are you finished writing? If you're not, let me know, please. And I'll go back. All right, so we're learning about the structure of the
all
leaf.
shapes and sizes, and their leaves.
Wait, let me show you this one instead.
Just like in animals, plants have different levels of organization, with lots of similar cells combining to form tissues, different types of tissues combining to form organs, and then multiple organs combining to form organ systems. To see how these fit together, we're going to take a look at the structure of a leaf, which itself is an organ. And along with the stem and roots, it forms an organ system that's function is to transport substances around the plant. This image here is a cross-section of a leaf, as though you sliced it from top to bottom and are looking inside. Before we explore these structures though, it's worth considering its function. Leaves are the site of photosynthesis, which we'll cover in another video, but basically it requires lots of carbon dioxide and water in order to make sugars. The water comes from the soil and is transported to the leaves via the roots and xylem, as we'll see in the next video. But the carbon dioxide diffuses directly into the leaf from the outside air, through lots of little holes called stomata. In fact, each of the leaves will have thousands of these stomata, and they're scattered through the lower epidermis, which is a layer of epidermal tissue. Once the carbon dioxide has diffused through these little holes, it moves up the leaf and enters the spongy mesophyll tissue, which has lots of air gaps between the cells, so that the gas can easily diffuse through to the next layer, which we call the palisade mesophyll layer. This is where most of the photosynthesis happens, and so the palisade cells are packed full of chloroplasts. Above this, we have the upper epidermis, which is another layer of epidermal tissue. These cells are almost transparent though, as the sunlight needs to be able to pass through them to get to the chloroplasts in the palisade cells beneath. And once photosynthesis has finally produced the sugar molecules, they're carried to the rest of the plant by these green tubes here, which we call the phloem. Now the main problem that leaves face is water loss. The xylem, which runs next to the phloem here, continually brings water up from the roots for the palisade cells to use in photosynthesis. but that water can be lost from both the top and bottom of the leaf. To reduce this water loss, the leaf has a waxy cuticle on top, which is basically a thin, waterproof layer of lipids that the water can't get through. When it comes to the bottom of the leaf, the main problem is the stomata, as the water will diffuse out of any gaps really easily. The leaves need these holes though, so they can get the carbon dioxide that they need. As a compromise, the leaves keep their stomata open for as short a time as possible, so that they can maximize carbon dioxide absorption, but minimize water loss. In order to achieve this delicate balance, each stoma, which is what we call a single stomata, is formed from the gap between two guard cells. When the plant has lots of water, so it doesn't need to worry so much about conserving it, the guard cells will be well hydrated, which we call turgid, and this makes the gap between them larger, allowing more carbon dioxide to diffuse through. On the other hand, when the plant is short of water, the guard cells will lose water due to osmosis, and they'll become flaccid. This in effect closes the stomata, meaning that the plant no longer takes in carbon dioxide, but more importantly, it will conserve its water vapor. Another adaptation is that the guard cells are sensitive to light, so they close at nighttime when photosynthesis isn't taking place, and they don't need carbon dioxide. And the reason that most of the stomata are on the underside of the leaves, rather than the top, is that the lower surface is more shaded, which makes it cooler, and so it means that less water will evaporate. One last tissue we need to mention is meristem tissue, which is basically the plant form of stem cells. They're found at the growing tips of the roots and shoots, and can differentiate into loads of different cell types, so that the plant can grow. If you haven't heard yet, you can find.
So we have seen now the parts of the leaf that we learned about. So let's just have a look at a video about transport plants. Then I'll give you a short break, and in the second half, we're going to get into the detail.
We could just say movement of water and minerals and movement of glucose and amino acids, but this is science, and so we like to have special terms to describe these processes. In this three-part video, we are going to look at the transport systems in plants for moving food, water and minerals around. We have a beating heart and circulating blood, but what do plants do? Cut a plant open and it doesn't bleed, so what happens instead? Plants have their own systems. They have a xylem, which moves water and solutes from the roots to the leaves, and a phloem, which moves glucose made in the leaves by photosynthesis and amino acids to the rest of the plant. Here are the xylem, and here are the phloem. Notice how the arrangement is different in the stem and the roots. The xylem and the phloem are found in groups called vascular bundles, and the position of these bundles changes for different parts of the plant. Both the xylem and the phloem are made up of rows of cells that form a continuous tube running the whole length of the plant. The xylem vessels are made of elongated dead cells that are impermeable to water and have walls containing lignin, a woody material. Because of this, xylem vessels are tough, which is why the vascular bundles in the roots are in the center. They help prevent the plant being pulled out of the ground. They are also more protected in the center. Whereas the stem has to resist being squashed and bent, and so it has the vascular bundles nearer to the edge to give the stem strength and support. The phloem vessels are made up of living cells. They transport sucrose and amino acids up and down the plant, depending upon where they are needed. Whereas in the xylem, the movement is just one way, from the roots up to the leaves. So we know that water and minerals go up the xylem, and amino acids and glucose go up and down the phloem. But how? In the second part of this video, we are going to look at the xylem and transpiration.
So that's exactly what we're going to do in the second half of the class. We are going to be looking at the xylem vessels. All right, so now that we have an idea of what we're trying to transport in the plant, which is the main thing is water for now. We're gonna be learning that in greater detail. Right? So I'm gonna give you a short break. We'll continue to the break. So we're looking now at water transport. As we said, that was the main challenge that we had for our leaves in order to do photosynthesis, is to get the water to them. So we're going to start exploring exactly how do we get the water from the soil to our leaves. So water is found in the soil. And the first thing is that water has to enter root hair cells. So of course, I'm going to be showing you a bit about the roots as well. We learned about the structure of the leaf, so we learned about the roots. So water enters the root hair cells by osmosis. Then water has to travel across the root by osmosis before it enters the xylem vessels that are in the roots. Then the water has to travel up the xylem vessels from the root through the stem until it gets to the vascular bundle in the leaf. Once the water gets to the vascular bundle in the leaf, that's when water can leave from the xylem vessels and go to the mesophyll cells. And the majority of the water that actually went up to the leaf is actually going to be lost from the plant by passing through the stomata by diffusion. So a good portion of the water that actually goes up there will actually not be used by the leaf, will just pass right out through the stomata. But that is actually very important. We actually do need to lose the water through the stomata to actually keep water to keep pulling water up the stem. So I'm going to give you a chance to make note of this. And I'm going to teach you some more about the xylem vessels, the special vessels that transport the water. So the loss of water through the stomata is known as transpiration, and that is actually important. That is important to pull the water up the xylem vessel against gravity without a pump. So transpiration, the loss of water through the stomata, is a very important process, which we'll explore further. So guys, I'm going to teach you now about the structure of the xylem vessel. Because they're the ones that are going to be transporting the water up from the roots to the leaves. When you're finished writing this, I'm going to actually show you a clip of the xylem vessel first, a video of it, and then we make a note of it. Another minute. Then I want to show you a clip on these xylem vessels, which are these specially adapted vessels. Right, let's go to look at this, let's visualize it, and then we write about it after.
Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe the structure and functions of xylem vessels and xylem fibers. In the last video, we saw that we find both xylem and phloem in the vascular bundles of plants. And if you haven't seen that video, then you should watch it now. In this video, we're looking at the structure and functions of xylem. Now, the first key idea you need to understand is that xylem actually consists of two main types of tissue. These are called xylem vessels and xylem fibers. We're going to start by looking at xylem vessels. Xylem vessels start as a series of plant cells running up the stem from the roots to the leaves.
So let me just pause right there. So basically, when the xylem vessel was being formed, what happened is that cells just became joined to each other. So you had a stack of cells joined to each other. But then, in order to convert it into a tube, all these partitioning walls had to be removed. So all the walls that separated one cell from the next had to be removed, and all the contents of the cell also had to be removed so that it could be converted into a hollow tube so that water could flow through it.
A series of plant cells running up the stem from the roots to the leaves. At a certain point, the carbohydrate lignin forms within the cell walls. Lignin is impermeable and prevents substances from passing through the cell wall. The living contents of the cells die, and the end walls between the cells break down. However, regions of the cell wall remain free of lignin. These are called pits, and these allow water and dissolved substances to pass between vessels. So the final xylem vessels consist of non-living hollow tubes, and the function of xylem vessels is to carry water and dissolved minerals up the plant from the roots to the leaves. We'll be looking at how this movement of water takes place in a later video. Now, if a xylem vessel is blocked or damaged, then water can move through the pit to different vessels. The pits also allow water to move out of the xylem, for example to cells in the leaves. Okay, I'm showing you some xylem vessels here with the lignin shown in red. Lignin can be arranged in spirals or rings, such as the ones shown here. In other xylem vessels, lignin can be essentially continuous, apart from the pits. Now this lignin helps to support the structure of the xylem vessel. When water is pulled up the xylem vessels, this causes the pressure in the vessels to fall slightly, and the lignin in the vessel wall helps to prevent the vessels from collapsing. Now, as we said before, xylem consists of xylem vessels and xylem fibers. Xylem fibers are formed from long, narrow cells. Very large amounts of lignin form in these cells, and just like in xylem vessels, the interior contents of the cells die. However, unlike in xylem vessels, xylem fibers are not used to transport water. Instead, xylem fibers provide mechanical support for the plant. So as we've seen, both xylem vessels and xylem fibers are non-living tissue. However, xylem also contains parenchyma cells, and we saw these in the last video. In the xylem, parenchyma cells can act as a store of starch. Parenchyma cells can also contain tannins, which are bitter compounds that deter herbivores from eating the plant. In the next video, we look at the structure and function of phloem.
So we saw that the xylem vessels are these dead vessels, right? So they initially were alive. So they were initially made from plant cells that were stacked upon each other. But as we saw in the video, the partition between the cells got removed and all the contents of the cell got removed so that it became hollow, so that it allows a passage for water to pass through. So xylem vessels are made up of dead cells that are arranged end to end to form long hollow tubes that transport water and minerals from the roots to the leaves of a plant. All right, so you can go ahead and make note of this. And remember that in the walls of the xylem, it has been reinforced with a substance known as lignin, right? So that the walls are even stronger. So it transports water and minerals. There are no end walls between the cells, and it only allows one-way flow. So water can only flow in one direction in the xylem vessels, right? So I'm going to give you a chance to copy this. So now that we have established what the xylem vessels are, we're going to be looking at a process I mentioned to you earlier, which is the process of transpiration, right? So the main process that causes water to be pulled up the xylem vessels from the roots to the stem is transpiration. So I'm going to spend some time talking more about this process here of transpiration, which is the main process that's going to get us to pull the water up the xylem vessel. Just like what you see in the diagram, you see water going up the xylem vessel. We're going to talk about this process of transpiration that pulls that water up against gravity without a pump. Right? So it's a very, very important process to discuss. Transpiration is described as the loss of water vapor from plant leaves through the stomata in the lower epidermis. The loss of the water molecules from the leaf creates a suction force called the transpiration pull. Or transpirational stream. Pulls more water into the plant and up the xylem vessels. So what the plants have done is that they have come up with a simpler, more effective way than using a pump. So instead of pushing the water up, what the plant does is pulls the water up. It uses a suction force to pull the water up the plant. Very brilliant, in fact, right? So because there are two ways you can get, if you had any object that you're trying to get up to a certain height, you have two ways to do it. You either push it up or pull it up. So the plants, if the plant were pushing up the water, it would have needed a pump. Now to bypass it uses the opposite way, which is it pulls the water up by using a suction force or a stream. And that suction force is actually by allowing water to be lost from the leaf. So that's very creative. By allowing the water to be lost from the leaf, that actually creates a suction force to pull more water in. So that is transpiration or the transpirational pull. which we're going to be exploring further.
Excuse me, could you go back to the first diagram, one before this one?
Okay, this one.
Yes, thank you.
I'll give you some time to finish it because we have one very important diagram coming up next, where we're gonna see this brilliant design that God gave plants. They could lose water in order to pull more water in. That's really what the plant does. So it seems counterproductive that you're losing water to gain water, but the truth is that the plant doesn't really need a lot of water for photosynthesis. So it can actually lose a lot of the water that comes in and still gets enough to carry out photosynthesis. I'm gonna give it some more time, guys, because I'm gonna have to completely stroll away from this to put the other diagram that we're gonna look at. This transpirational pool or this transpirational stream. So let me give everybody some more time to finish completely writing it off. So let me just give it some more time. And then we're going to explore the transpirational stream in greater detail. And when we understand this part of it, when we understand the transpirational stream, we can see that if we disrupt the stream at any point, then water will stop flowing in. So it means that if the leaf close the stomata, so those are the pores that are in the lower surface. If those pores should ever close, and then water cannot be lost from the plant, then it also means that water cannot be pulled into the plant either, right? So, which is strange. So if for any reason the plant has to close the stomata, that's going to stop the plant from being able to pull water in from the soil. And you might wonder then, why would the plant close those pores? There are times when it has to close it. Sometimes it actually is losing too much water. It's good to lose water, but sometimes you're losing too much and the plant might actually be wilting. Or maybe there isn't a lot of water available in the soil. So maybe in the case of a drought, right? So if there is not a lot of water in the soil, the plant might want to close its stomata to prevent itself from losing too much water so it doesn't wilt, right? So those would be some reasons why the stomata will close. Also, like in the night, in the nights when the plant is not doing much photosynthesis, it might close or not have as many stomata open as well. All right, so I'm hoping that we got all of this note now, because I'm going to completely scroll away from it to display the next diagram. So I'm just checking before I scroll. If anyone is still writing. So this is now what we're going to be looking at now, the transpirational uphold. All right, so I'm going to walk you through it. You can do like a rough sketch, guys. The reason why I like giving you things to sketch, diagrams, is that it illustrates the points better. And it also allows you to include them in your answers. And that's one of the reasons why we give you past papers to do every week. Because we want you to immediately see, once you learn something, we want you to immediately look at the questions that are asked. And so it allows you to start formulating your answers. I don't want it to be that when you get in the exam the first time you're asked, right? I want to condition you from no. So I provide you with these diagrams so that you can include them in your answers. Because when you're answering your CXC questions, even though they give you a bunch of lines, they are watching to see if you're going to just present one long paragraph, or are you going to spend time and organize it into points and even add illustrative diagrams. When a student does that, that demonstrates mastery to the examiner. Versus when you just sit down and you're just rambling and just write one long paragraph and the examiner has to be sitting down there and trying to figure out how many points are you really trying to make, right? So that's one of the things that I'm really trying to train you for here, which is how do you present yourself as a student? All right, so here is the transpirational stream. As I said, the diagram is there to illustrate the point. And I did it like this because I actually, maybe you can't see the numbers that clearly, but number 1 is actually right here. And I wouldn't necessarily tell you that you have to draw this diagram right now. This is mostly for illustration, all right? So the first way that this stream starts, if you were to tell what was the first step is really when water vapor is leaving from the leaf through the stomata, all right? Water vapor, that's the first point right here. Water vapor comes out. Now, the water vapor that comes out has to be replaced. So basically, I want you to think about the water molecules. I want you to imagine that the water molecules are all linked up together like this. They all are linked. So it's basically like they form this column. So essentially, when we lose water molecules from the leaf, then water has to be pulled. So this is point number 2 right here. So to replace, so the water that we are losing were in the air spaces right here. So just pretend that we had water molecules in the air spaces and they are leaving. So now that we have lost water molecule from the air spaces, we must replace the ones that were lost. So to replace the ones that were lost, we take water from the cells, like the spongy mesophyll cell, and water from the spongy mesophyll will replace the water that we just took out of the air space. So now that the spongy mesophyll cell, they are losing water, so the water that they lost must be replaced. So they get back their water from the xylem vessel. So this represents the xylem vessel. And some of the water also goes to the palisade mesophyll. So anyway, because we are now pulling water from the xylem vessel to replace the water that we just lost from the cells, then that's what creates that suction force. So every time we pull water out of the xylem vessel at the top, it creates a suction force to pull more water up the xylem vessel. So now we end up have water being pulled up. That's number 4. Then number 5 So because we have been losing water from the xylem vessel, we have to pull water from the cells in the root to replace the water that we took out of the xylem vessel. And then because we are taking water from the cells in the root, we pull water from the root, well, from the soil to replace the water that we lost from the root. So it's literally just this continuous chain. So just by losing water from the through the stomata, we replace that by pulling water from the spongy cells. Then the spongy cells pull water from the xylem to replace what they lost. And then that water that we are pulling from the xylem causes a suction force to pull the water up, and it pulls water from these cells root to replace what the xylem lost. And then the root is losing water, so they now have to pull water from the soil to replace what they lost. And that's why it's called a transpirational stream, transpirational pull, which really starts off by losing water vapor through the stomata in the leaf. Okay? So basically what I'm saying is that if you should close the stomata and you cannot lose water from the pore, then the entire stream gets stopped. Okay? So what you need to copy are the steps, not the diagram. Okay? The diagram is just to illustrate what I was saying here. So the steps are, so we say the transpirational pull or stream pulls water up against gravity, right? And it starts off when there is a diffusion of water vapor out through the stomata. Notice I said water vapor, not liquid water. So the water has to be turned into a vapor when it gets two places. So in the air space, the water is not liquid, it's vapor. And when it's coming out, it is vapor. Everywhere else, it is liquid water, right? So there is the diffusion of the water vapor out through the stomata. Then there has to be evaporation of water from the spongy cells into the air spaces. I should have mentioned that the water was coming from the air spaces right here. So remember, you don't need to copy the diagram, guys. The diagram was there for illustrative purposes. So the first set of water, we lost the water vapor through the stomata from the air spaces. And then we had to replace that water. So we had evaporation of water from the spongy mesophyll cells into the air spaces to replace what was lost. Then water gets pulled from the top of the xylem vessels to replace the water that was lost from the spongy mesophyll. Then that causes a column of water to be pulled up the xylem in the stem. Then water enters the bottom of the xylem located in the roots to replace the water lost at the top. And then water gets pulled from the soil into the root hair cells, right? To replace what the root hair cells would have lost, okay? So that's a summarized version of the transpirational pull. And then I'm going to show you an experiment that you need to know of, and that's going to come in past papers. That proves to us that really water is traveling with the xylem vessels. So go ahead and make note of the steps, please. Yes. When we say evaporate, it means that it's converted from liquid to vapor. How does it replace what was lost? Because remember I was saying that the water molecules are they're like joined together, like a stream. So when some of them are leaving, it automatically pulls on other molecules which are beside it. Well, remember that if water molecules were lost, right? So for example, we had 100 molecules of water that went out, right? Then it means that these 100 molecules were in this space right here. And these 100 molecules that were in this space came out. So now 100 molecules of water need to go back into the space. So those molecules are going to come from the cells that are surrounding the space. So 100 water molecules that were in the cells are now going to have to come out and go into the space. And now the cells are short of 100 water molecules, so they're going to take those 100 molecules from the xylem vessel, right? And then the 100 molecules that we took from the xylem vessel has to be replaced. So we're going to take 100 molecules from these cortex cells to go back into the xylem. And then the cortex cells now lost 100 molecules, so they're going to pull that from the soil. Yeah. Yeah, that's it. So we said that if the stomata should close and water is not leaving, then there would be no pull to draw any additional water into the plant. Right? So you might then ask yourself, then what's the whole point of trying to bring water up to the leaf if all we're going to do is lose it? But this is the reality. So if for example, we are losing 100 molecules of water and we're pulling up another 100, in fact, we did use some of the water. Notice that some of the water is going to the palisade cells. So that's the real purpose of all of this. The whole purpose of the transpirational stream was just to bring water and so that it would get to the palisade cells because they're the ones that are really doing photosynthesis. But it's just that they don't really need much water. So they might need probably, just hypothetically speaking, 3 molecules in order to do photosynthesis. But for them to get the 3 that they need, we have to lose like 100 to pull up, to keep water coming in. So that's just a hypothetical situation. But we're saying that the majority of the water that is coming in is actually going to be lost, but a small fraction is actually going to be used in the process of photosynthesis to make sugar. So essentially, yes, all of this is happening so that the palisade cells can get the water that they need. But we are saying that why this stream is important is because plants can reach up to very high levels. They can be very tall as 3-story buildings, and leaves are at the top. So how are we going to get water from the soil to get to those leaves at the top? And there is no pumping system in the plant. So if you don't have any pump to push the water up, then you have to devise a second system where you're going to have to pull the water up by a suction system. And so this is the suction system that plants have devised. And the plants don't only use water for photosynthesis. Some of the water is actually going to be stored in the cell to allow the cell to be swollen and turgid. And that allows the plant to be upright. So some of the water actually stays in the cells to keep the cells swollen so that the plant can stay upright. I don't know if you've ever seen a plant that has wilted. Have you ever seen a plant on a hot day that it just falls over and it's no longer upright? No, it's not upright. When it wilts, I'm going to find a picture. You'll more likely see that with like plants like callaloo or something like that. You're not going to see a tree, but like I'm going to show you. So copy this and I'll show you what happened with the wills. So what I did right here, I'm just showing you what I mean when I say to you a plant wilts. All right, so I'm sure everybody would have seen that. So we're saying this is what happens that when the plant loses too much water, and a lot of the water, some of the water that it's taking in is actually to stay in the cells and keep them swollen so that the plant can be upright. The plant loses too much water and the cells are no longer swollen, that's when it bends over like this, wilting, it's losing too much water. And what it is losing, it's not able to get it back from the soil, right? So if there is like a drought or something, and there's not enough water in the soil, then the plant might risk losing more than it can get back from the soil. And that's when we wilt. Which is why we water plants, yes. Right, so water transport is not only important for photosynthesis, but it also is important for keeping the plant turgid or keeping the plant upright by keeping the cells swollen. All right, so I'm going to give you a chance to finish this, like 2 more minutes. And then I'm going to look at the classwork and guide you through it. You know, how we can, based on what we've learned today, how can CXC ask us questions on this? And there's one critical experiment, but there's a handout I'm going to discuss with you. So I know you've done a lot of writing. So what I did is that I put some of the notes in a handout. All right, so what I'm going to do, I'm going to just make a note, C handout for celery experiments. So sometimes, guys, whenever you have a handout, in order for you to know or to remember to look for the handout, I normally ask you to write a note in your notes, right? So that when you're going back through your notes, you can see where there is actually a handout you should look for, all right? So that's usually what I do. So there's a handout I'm going to discuss with you, and you're going to be looking at, we're going to be discussing the experiment with the celery stick, and you know that you should look up for the handout to get that experiment. So we kind of like to do experiments with celery because it kind of has a transparent stem. So we can actually add dyes to water and we can actually see the water move through the celery. So I'm going to actually show you that experiment. All right, so there's this experiment that CXC likes you to know, which is basically us proving that the xylem vessels are the ones that carry water. So in the big trees, we can't see the vessels in the big trees, but in a plant like xylem, sorry, like celery or callaloo, where the stem is transparent. If you added some dye to the water, you could actually see the dye moving through the plant, and you definitely be able to see that yes, it travels in the xylem. So let me show you this experiment, and then we go to the handout.
Hi, and welcome to Reuben H. Fleet Science Center Experiment of the Month. My name is Laura, and today we're going to be doing a celery and food coloring experiment. It's a pretty simple experiment you can do at home to learn a little bit more about plants and how they absorb water. So first, let's take a look at what supplies we'll need. So, we'll need celery, water, food coloring, cups, and scissors. Have all your supplies together? Okay, great, let's get started. First, we're going to add our food coloring to our jars of water. You can add them in any order into any jars that you want. Put about 10 drops into each one. Okay, just a couple more drops and then we'll be ready to go. Nice, now we have our colors all mixed. Okay, let's grab our celery stalks now. Pick 4 stalks that have a lot of leaves at the top. Before we stick the celery into our jars, let's cut the bottoms off to help them absorb water better, just like we would do with a bouquet of flowers at home. Now let's put our celery into the jars. And you can stir the water around a little bit to make sure the food coloring is all mixed in. Now let's wait and watch to see what happens. What do you think will happen? Discuss this with your friends while we wait. Just like you and me, plants need water to survive. But where do plants get this water? Water comes from the soil surrounding the roots. Water is taken up through the roots into the trunk of the tree through special cells called xylem. Xylem cells are long rectangular cells that are packed close together so water can easily move through them. Xylem lines the tree all the way from the bottom roots up to the top branches. So water moves from the roots, up the trunk, into the top branches, into the leaves. In the leaves, water is used for a process called photosynthesis. That's how plants make energy. Water also exits through the leaves in tiny pores called stomata. This process is called transpiration. Because of photosynthesis and transpiration, there is less water at the top of the tree than at the roots. This causes water to be pulled in at the roots and up the trunk of the tree to the leaves. This is a natural process whereby water is moved from an area of high concentration, or where there's a lot of it, to an area of low concentration, or where there's less of it. So that's how plants absorb water. Now that we've learned the science behind it, let's check on our celery. We left our celery in the colored water for about an hour and a half, but you can leave it in as long as you'd like. The longer the celery is in the water, the more color it'll absorb. Now let's take a closer look and see what happened to our celery. Oh my gosh, look at all those colors. It's easy to see that water was pulled up through the base of each celery stalk. Now let's check out the tops and see what they look like. As we move from the base of the celery to the top, you can see red coloring throughout the stalk. Even in the leaves, there's red coloring. That's pretty neat. It's just like our tree. For a good comparison, take an unused piece of celery and look at the difference. Now let's check out what our celery looks like on the inside. Take a closer look.
You can actually see the food coloring, and the ones that are colored are the xylem vessels. So this is a proof to prove that this is where water travels.
Look at the fibers in the celery. That's the xylem, where the water was absorbed. Our are celery and food coloring?
So because of the fact that they are transparent, it makes it easier for us to see. So this is a classic experiment that you are expected to know, all right? So I'm going to show you I'm going to show you the handout now. So the handout is going to be in Canvas. With the xylem is always that large. So yeah, but it's not, what you're seeing is not one xylem, you know, it's hundreds of them joined together to create the xylem fibers. Because remember that the xylem was made from cells that were joined together. Cells are not able, you're not able to see cells with your naked eye. So the fact that you were able to see the xylem with the naked eye means that you have millions of cells that are there joined together for you to actually be seeing it. So when you, for the areas of the plant where you did not see the color, it could have been the xylem fibers or it could be flowing vessels. So there are other tissues inside of the stem so those would not have been colored. All right, so guys, we just started this module, transporting plants. All right, so the notes that I'm going to be looking at right now, it says week 7 class notes. All right, I didn't have it there, sorry. But it will definitely be, usually when I have handouts, I normally put it with the classwork. Sometimes put it by itself, but apparently I never did that. So it will be with the classwork, week 7 classwork. So if we open the week 7 classwork, then we will see several things. So we're going to see the classwork down here, which I'll discuss shortly. And then there's a tutorial video. So as many times I do have a video that I explain how to do the past paper. If you don't remember, sometimes the videos you can watch it again. That's where I say, okay, this is how you answer the past paper. But anyway, this is the handout. So I'm going to download it and then go through it. So some of what is in the handout, we would have already covered and we would have already written notes on it. So I'm gonna skip. All right, so I hope guys at home you're seeing the handout. So the first part of it was basically talking about the process of the transpirational pull, but we already but this has a little bit more background information than I told you to write. It's basically saying that it is a process by which water vapor is lost, and the loss of water creates a stream that pulls on more water into the plant, and water molecules are able to bond with each other by a process known as cohesion, and so it allows water to act like a column, and then it describes the very process we already discussed. All right. So now I'm skipping down to the part about the experiments for transpiration. So celery and food dye, which is what we just saw in the video, it proves that water travels in xylem vessels. So this is the experiment. So you put the celery in the dye, and after a while you take it out, you're going to see a column with the color of the dye all the way up to the leaves, right? And if you should take out the celery and cut it across, you would notice that there are specific tubes that got colored. Notice there's no color in the center. And this is showing you exactly where the xylem vessels are found in the stem, right? So a ruler can be used to measure the distance traveled by the food following the time, and that tells you the rate of transpiration. The experiment can be set up with a fan beside of the celery to see the effect of wind speed. So if you did this experiment without a fan and you use a ruler, you gave it some time, maybe 10 minutes or whatever time, and you see what distance the dye went, then that would be distance divided by time, gives you rate. So if you decided to put a fan in front of the celery and do the same thing, then you will probably notice that it travels faster. The water is going to travel faster up the xylem when you put a fan in front of the celery. Next week, I'll explain to you why, right? So another way that you can actually prove, another experiment to prove transpiration occurs in a plant is that you could actually get a potted plant and cover it with a plastic bag. And after some time, you're going to see droplets of water in the plastic bag, right? So the only way droplets of water could reach in the plastic bag is that water must have been evaporating from the leaves and then it collects in the bag. So this is another way to prove that transpiration occurs. Now, another way, another experiment that could be used to prove that the leaves are losing water vapor is by actually getting a potted plant and putting the bag around the root of the plant, right? So you cover the root, not the leaves, and you leave the plant on a scale. Now, you're covering the roots because you don't want any water to evaporate from the roots because there is water in the soil. And you want to prove to anybody that any water that is going to be lost has to be leaving from the leaves and not from the soil. So you cover it and you leave the plant on a scale and you give it time and you're going to notice that the mass of the plant is going to fall because water vapor will be leaving, so the mass of the plant will fall. So basically, these are different experiments that can be used to investigate transpiration, right? So you could set up the apparatus as shown, and the initial mass of the plant can be noted. The bag prevents loss of water from the soil by evaporation. You could measure the mass after every 1 hour and record in a table. And the decrease in the mass with time gives the transpiration rate. And you can plot a graph of change in mass versus time, and the gradient gives the transpiration rate, right? So the rest of the handout, guys, has some additional notes about the structure of the leaf, which we already wrote that. It shows you the leaf. So a lot of this is just additional notes, right? I always like giving you extra reading. So that's the handout. So now that we have looked at that, we're going to go back now to the classwork and see what we have to do. So the week 7 classwork would have the handouts, has a tutorial video, and then the past papers. So let's see now what you can be asked in CXC based on what we covered today. All right. So the first one says, and this is from 2019, Figure 1 shows the apparatus that was set up to measure the rate of transpiration of a potted plant located in a room beside an open window over a 12-hour period. Describe the procedure that uses the apparatus shown in Figure 1 to investigate the rate of transpiration of a potted plant within the 12-hour period. All right, so literally, guys, the handout that I just looked at this very same experiment and gave you the procedure. So that you'd actually get the answer for this by looking at the handout. See that you'd get the mass of the plant, make sure you cover it, and take the mass every 1 hour. So that's what they mean, right? So you can look back at the handout to get the answer for this. The next one, 2014 question 2C to D, shows you a section of the stem of a flowering plant. So there are several tubes in the stem. The important thing is, can you identify which one is the xylem? So the xylem one, guys, is would be vessel P, right? Vessel P. Because this one is hollow. The partition between the cells is almost gone, and it has water traveling there. So you might wonder, okay, so why isn't vessel Q the xylem? Vessel Q, it kind of confuses you to look like the xylem, but it's not, because it has a cell beside it, a living cell. So the xylem vessels are completely dead. The phloem vessels are not fully dead because they actually have cells that are beside the tubes. And that's why I would have known that Q would not be the xylem, because I'm seeing cells beside the tubes. For the xylem, there are no cells beside them, just columns of dead cells. So vessel P would represent the xylem and Q would represent the phloem. Then explain why plants do not need an organ like the heart in their transport system. They don't have a pump. Why? I told you that earlier in the class. I gave you that in the earliest notes. I said they don't have a pump because they are not as complex as humans. They don't carry out as many processes as humans, right? So that's why they don't need a pump. It's 2 marks. So remember, the amount of what you write depends on how many marks it's worth, right? So, and then they said, can you give one mechanism by which plants move their fluids through their transport vessels since they don't have a home? This is where you'd say they use a transpirational flow, right? So 2 marks. The next one now, the next question is based on the celery experiment now. So this is 2013. So in flowering plants, xylem vessels transport substances. Draw and annotate a diagram to show how the following materials could be set up to investigate the rate at which water moves through the xylem vessels under 2 different conditions. You're given 2 beakers, water, dye, stopwatch, fan, ruler, plants with transparent stems. And you're going to tell them how you're going to show one plant, right? Just like exactly the handout, right? You're literally going to put a celery stick in the dye, water in the beaker which has dye, and you're going to leave it and start the stopwatch, and probably after a few minutes, probably 10-15 minutes, you measure with a ruler how far the dye went. And then you have distance divided by your time. Then you're going to set up the second one with the celery stick in the dye, but you're going to put a fan beside it. You're going to give it the same amount of time, and then you're going to stop and measure with a ruler how far the dye went, and then you divide it by time. So that will be how you'd be able to compare how water moves up a plant with or without a fan. So that's what you're going to describe. And of course, you include diagrams. Write a suitable hypothesis for this experiment. So a hypothesis is like a question that you're trying to answer. Or so anytime you do an experiment, guys, anytime you do an experiment, there must be something, there must be a reason why you're doing it. So there is some statement that you're trying to investigate. So the fact that we were doing an experiment with a celery stick with the fan and one without the fan, what we were really trying to probably understand is, does a high wind speed cause the transpiration rate to be higher? So that more than likely is our statement, and we're setting up this experiment to investigate. So I actually gave you the answer for that. Relate the structure of the xylem vessel to their role of water transport in plants. So the structure of the xylem vessel is that they're dead hollow cells. And that's good, but they're dead and hollow. It means that the water doesn't have much resistance. As if there were things inside of the xylem vessel, the water would have to be pushing past the contents of the cell. That would make it harder for water to move up. So the fact that they're dead and hollow means that there's less resistance for the water. And also the fact that we have lignin in the walls of the xylem vessel reinforces them so they don't collapse, right? So the xylem vessels don't collapse when the pressure of water is high inside of them, or even when the pressure is low, it doesn't collapse. The tubes will stay open. So those are 2 features you could say why to relate the structure of the xylem to its function. Then finally here we have this section of a dicotyledonous leaf. Name one function for which the leaf is suited and how is it best adapted for that function. Photosynthesis, I would say, be one of the first things. You should say why the leaf, what the leaf does and how it's best adapted. You can give anything. You can say it has polysaccharide cells that have a lot of chloroplast. You can take any feature of the leaf and say why it's well adapted, right? Use 3 arrows only to show what path water will take when diffusing out of the leaf. 3 arrows. So I would say you want to show water leaving from the xylem into the airspace, right? Although that's like when traveling through the airspace and then coming out, right? So you could do that, 3 arrows. And then identify the structures labeled 1 and 2. So this is why I know 1 is the xylem clearly, and you must figure out what number 2 is. Remember, you already drew the leaf. So there's no part of the leaf that somebody should ask you and you don't know. Name one consequence to a herbaceous plant, like the celery, if there's a temporary unavailability of water to replace the water that was lost from the leaves. So remember that diagram I showed you earlier? What did I say would happen if a plant loses more water than it can absorb? It wilts. It doesn't die, it wilts. All right, so it just falls over like what I showed you in the picture. So these are simple things, you know, guys. Explain how water from the soil gets to the leaf. This is where you're going to quickly tell about the transpirational stream, right? I know this is a lot of questions this week, but I really was just trying to get you familiar with the past papers surrounding this part of the topic. Explain how one structural feature of plant cells help in transported plants. The cell wall is one feature. The fact that the cell wall is permeable to water, water can freely pass through the cell wall. And finally, explain why all the water taken up in the roots of the plant is not lost by evaporation. So we said that all the water doesn't get lost because some of the water is actually going to be used for photosynthesis and some of the water is actually stored in the cells to keep them turgid. So that's why the plant doesn't lose every single drop of water that it absorbs. All right. So guys, yes, I know we have a few questions to do today. I'm going to actually stop now. But it looks like a lot, but when you actually sit down to do it, it's not that much. Because as I said, I answered this directly in the handout, the steps for this experiment. This one is just filling in this, right? Why Q is the phloem? Why doesn't the plant need a pump? Because it's not as complex, doesn't do as many reactions. One mechanism, we said transpirational pull. So I'm already giving you most of the answers. This is the one with the celery. You're gonna draw one celery in a beaker with colored water, one in the other one with a fan, one without a fan. Right? And that's what I say, draw and annotate the diagram. Then, two features of the xylem that makes them suitable for water transport. We say that they are hollow, dead, so that the water can flow easily. The walls are reinforced with lignin, so that the vessels don't collapse, right? Then they ask you to say what the leaf is adapted for. Photosynthesis, how is it adapted? You can select any adaptation you want. Put 3 arrows to show how water will come out. Water structure 1 and 2. And one consequence if a plant uses too much water. How does water move from the soil to the leaf? And one feature of the plant cell that helps to transport cell walls, which are permeable to water. Water can freely pass the cell wall. And why is it that all the water taken up by the roots is not lost? Because some of the water is used for photosynthesis and some of the water stays in the cells to keep them turgid. Literally, those are all the answers for the past papers, right? You might wonder, why am I giving you the answers and still asking you to write it? Because the process of you even writing back the answers is actually making you learn, right? So that's why I still want you to do it and submit it so we can mark it and grade it. So this is where you're going to summarize the transpirational pull, right? So in the transpirational pull, part C, I gave you like 6 steps. But this is a 3-mark question, so you have to be using a judgment. If it were 6 marks, I would have written back exactly what I gave you in the notes. But for 3 marks, just make the points shorter. Right? Those 6 steps, just make them shorter. And that should be it. All right? So guys, we're going to stop here. Remember that you normally get 2 days. So the classwork has to be done by Wednesday and submitted by Wednesday. So that we can mark it and return it to you. All right? Please ensure that it's done. We're going to stop here for tonight. I know you're tired. Enjoy the rest of your night, guys. We'll continue with transporting our plants in our next class. Any questions before we go? All right, guys, please do the questions when the information is fresh. Right? If you really could push and do it tonight, that would be good because literally you'll remember me saying the answers to you. When you delay and you don't do it tonight and you don't do it tomorrow, by the time Wednesday comes when you're hurrying, you would have forgotten a lot of what I did say. And I always usually tell you most of the answers. That if you did it right away, you just capture it in short-term memory, get it out of the way, and move on with life. All right? All right, so that's it, guys. Thank you very much for joining and enjoy the rest of your evening, everyone. Bye, guys.